Good morning. For this next session, it's now my pleasure to introduce Dr. Zandy Forbes, President and Chief Executive Officer from MeiraGTx. So the format for the session is roughly a 20 or 25-minute fireside chat. If you have any questions during the session, feel free to type them into the question box and we'll try to ask them on your behalf. So Zandy, thanks for joining us. To start off, would you give us an overview of Meira's manufacturing and CNC capabilities and also discuss how they fit into the company's broader strategy?
Yes, so we currently have a up and running GMP facility in London, which is flexible and scalable for commercial supply. And that was one of the first things we did when we formed Meira 2015, was embark on building this facility in collaboration with the regulatory agencies. That was important to us because we wanted to not only ensure that we could supply our clinical trials, but we wanted to ensure that as we got into later stage clinical trials, we would have control over the process and comparability and eventually the BLA filing for our products throughout their life cycle. So manufacturing was definitely a focus when we set up the company, and that was our first facility. In 2019, we embarked on a second facility, which is built in Shannon, Ireland. It is on the same sort of design, so very flexible, everything single purpose, single use. And each room has its own air handling, so it's like a mini facility. And in that facility, which can go to even larger scale, although it can still do small scale, we have a plasmid facility where we can now manufacture GMP plasmid. And in that facility, we have a large excess, actually, at the moment, of capacity for plasmid production, well outside what we use in-house. But it was very important to us to build a plasmid facility because we were seeing constraints in supply. And we are not only supplying clinical material for us, but through our agreement with Janssen, we are the clinical and commercial supplier of the programs that we work with Janssen on. So while we initiated our manufacturing strategy in the absence of a partner, it's really been accelerated and boosted by the needs for commercial supply. As you can imagine, we have a phase 3 study with Janssen, and that has involved CMC filings in many global jurisdictions based on our manufacturing process. So in addition to the facilities we have built over the last 7 years a platform process that can be used with many capsids, many vector genomes that we use in our facilities. And finally, right now, we understand there's supply chain issues, but as we move towards commercialization potentially, there is a huge capacity constraint in the QC for analytics. So in that site in Shannon, we're currently bringing in-house the QC for stability and release so that a potential commercial product could actually be released at a commercial scale. That's currently in the world. There isn't enough CRO capacity to do that for many commercial gene therapy products. So we've gone from plasmid production to the other end of the manufacturing to QC and release. We also have fill and finish. So it's pretty broad and comprehensive.
Got it. That makes sense. So in terms of the supply and the just capacity constraints that you were just alluding to, you mentioned the constraints regarding QC and analytics capabilities. Are there other constraints that you've noticed in the last couple of years, and has that pattern evolved from what you've seen?
I would say, well, initially, let's say 6 years ago, there was huge concern about capacity for viral vector manufacturing. how much vector can you make, and how high a quality is that viral vector, and are your facilities and your process something that regulatory agencies globally will accept as reliable, consistent, and producing consistently safe, efficacious material. So that was a big concern for some companies who use very large doses, the capacity for viral vector production. And that's addressed by the process and the facility that's GMP approved. However, I would say prior to 2020, we did recognize that if you are going to supply commercially and even a number of clinical products, there are delays in getting plasmid, and GMP appropriate plasmid was not readily available and still isn't readily available. So that was the first thing we built at Shannon. I would say over the last 2 years, as we're entering a pivotal, which is now ongoing, and we're starting to look at what one needs for commercialization, that's where the issues with QC and CROs have really become really, really critical. I mean, as an example, just an example is, I think last year, I'm going to say in the third quarter maybe, we went to a CRO, we were releasing a batch and we went to a CRO and they gave us, this was in the third quarter of 2021, they gave us a November 2022 date for a final report on one assay. That obviously is not acceptable. No one can delay release of material, even for a clinical trial, with that sort of delay. So we actually bought that particular assay in-house and we were able to release that material last year. So that's the sort of things we're looking at. It's not a few weeks, it's a really, really bad capacity issue. And it's an expertise issue as well, because for, as you're dealing with regulatory agencies for late stage products, for pivotal products, and potentially for commercialization, which you have to prepare for a BLA, there's a lot of these aren't your normal assays. They're vector specific or capsid specific or, cell specific. And it's complicated to not only develop the potency assays, to develop them, to make them cell-based, to make them reliable so that you can show your material is reliable. And at the moment, we just saw that as a huge risk from a time perspective. And by the way, plasmid and QC are the biggest part of the cost of goods. So by bringing it in-house, you vastly reduce how much you're actually paying on a particle per particle basis for your released material.
Interesting. That makes a lot of sense. And I think we'll talk a lot more about that on the upcoming panel at 12. And I think a couple of points that you talked about earlier with regard to the scalability of your process and your facilities. Can you discuss the scalability and the capacity that you currently have, and any kind of flexibility that you have for to meet future demands?
So our initial facility and our second facility in Shannon were built in discussions with the European regulatory agencies to be what they wanted to see for a viral vector facility of the future. And that involved a number of things. Number one is everything is single use. So the rooms in the facility are essentially empty, and then bioreactors are wheeled in, and the process, a run happens over the next 2 weeks or 3 weeks or however long it would be. So each room is completely changeable. It has its own air handling, can shut down, clean itself, and open up again a few days later, and a completely different viral vector using a different bioreactor, potentially a different size bioreactor, can be run in that same room. So with that single-use philosophy, you have a huge amount of flexibility. And that means that you can run different clinical batches in rooms batch after batch because they're totally cleared out, and then you will in the bioreactor. It also means that from one day to the next, you can increase the scale that you're manufacturing in that room, because you can increase from a 50 litre to a 500 litre, right? Just depending on what bioreactor you use. So the facilities are really scalable. In addition, the Irish facility can have even larger bioreactors. So from a 50 all the way up to a 2000. So that's extremely scalable, but still has that flexibility of being able to be used for what you need when you need it. That's the infrastructure was built for really being able to scale into the future without completely changing your facility and your process. The process is important because the process, we have a platform process that is both adapted to any capsid and vector that we use, and it is actually tweaked for every one, although very similar, and we continually work on that. And that process can be scaled in two ways. It can either be scaled by going from a 200 to 2,000 liters, right, in larger bioreactors, or you can take 10 200s and put them in a row. So it's incredibly flexible and scalable. The real point here was to really have a process and a facility that from IND all the way through to commercialization could supply your needs for phase one, a pivotal, all the regulatory documentation and potential BLA and commercialization without having to change the facility, the process, and because those changes for viral vectors are quite onerous with respect to showing the regulators that it is comparable. And I say comparable rather than identical, but there's a huge hurdle to be comparable in the eyes of regulatory agencies, and then reliably so in every batch. So yes, we thought about all of those things. And it's very important if you're a company with a pipeline of clinical programs and yet you want to develop them to commercialization, and we have a partner that we do that with. Johnson & Johnson's very experienced in not doing phase 3 studies, getting things approved. So when we did our partnership with J&J, Janssen, we learned a huge amount, not about manufacturing for the clinic, but what's needed for a BLA. And that's been really transformative for the manufacturing group at Meira.
Got it. So on that topic, you mentioned earlier that your plasmid capabilities can be applied towards your Janssen collaboration, and also how you've learned regarding what's needed to bring something to BLA and commercial stage from that. Are there other ways in which the manufacturing has played into the collaboration with Janssen?
I think from Janssen's perspective, when we did the collaboration, the fact that we did have expertise in CMC, which they at the time did not yet have for AAV, and that we had a facility so we could rapidly supply clinical material, was one of the reasons that they wanted to do the deal with us. And so it has been very important. And from our perspective, the collaboration has really allowed us to up our game, so to speak, to be ready for potential BLAs and potential approvals. So, we've learned a huge amount about later stage manufacturing. And I think one of the reasons Janssen's partnered with us is because at that time, they didn't have the expertise in AAV production, all the facilities. So it really has been a collaboration.
Mm-hmm. Right. Great. And so you touched a little bit regarding the process that you've developed, that it's compatible with different serotypes, et cetera. Can you expand a little bit on your process and whether there's particular innovations that you've implemented or you're looking at that could circumvent some of the cost drivers or challenges that have been seen in the space?
Yeah. So yes, we have made process developments, I'll call them, which are proprietary. And so we've obviously filed protection, and we actually at Meira have a group of, I think, about 30 people in process development. So we really look at the science of AAV production. We, being one of the leaders in the sort of development of a process from 2014, we work with a lot of the suppliers of transfection materials, bioreactors, thermometer, all of those things. And we often beta test stuff for different suppliers in the industry. So we are constantly involved in the innovation end of AAV manufacturing and vector manufacturing in general. There is no one thing that we focus on. I would say that as we continue, we use triple plasmid transfection, and there's many changes that have been made to increase percentage full, to make manufacturing more reliable, to increase yield that can be applied across the platform. We also have a group working on producer cell lines, on cell lines that have rep and cap, on how to regulate genes within manufacturing that can increase efficiency by looking at the cell biology of manufacturing. So when exactly do you need the cap proteins? When do you not need them? When does DNA replication occur? And I think you can have producer cell lines which you can control in a way that you will be able to get much higher yield, and importantly, a really much higher efficiency packaging. So that gives you higher yield and higher full packages. So we work on all of that. So we have a process which is constantly being improved. And we also have new technologies that we're applying to different processes like cell line development.
So maybe this relates to what you just said, or maybe this is separate, but you also have gene regulation capabilities that you're exploring within your pipeline. Can you discuss how that fits into your manufacturing, either in terms of assisting manufacturing or in terms of how you plan to address manufacturing for your gene regulation program?
So our gene regulation programs, all of our viral vectors that we've made, many viral vectors that we've made that are regulated by oral small molecules that have gone in vivo, those are manufactured in exactly the same way. There's no change in any of the vectors we've made by the presence or absence of the control cassette. And obviously from a very early stage, we test the manufacturability of all of our potential products. Now, we can even engineer double switches with different sequences so they can be manufactured. Now, it is a very interesting potential use of our gene regulation to do exactly what I've just said. So to be able to control cell behavior, real synthetic biology for the first time, that you can really precisely turn genes on to exactly the right level from zero. And you can imagine how you can use that in virus production or any manufacturing, frankly, if you understand exactly what factor you're adding. How can you do that genetically and precisely with a small molecule? So we do that, and we look with our regulation at cell therapy as well. So that's more in the manufacturing. So how do you control genes in cells? So that's manufacturing and cell therapy, and then the viral vectors, which we control. But our switch can be used really broadly, and there are lots of ways that, controlling cell behavior can be incredibly useful in increasing efficiency of manufacturing in general.
Got it. That's really interesting. And so moving from that, you touched on this a little bit, but what do you currently see as the biggest CMC challenge in gene therapy right now? And are there specific solutions on the horizon?
The biggest challenge right now, QC. I've already said that twice, so I won't go on again. But yes, and it's really been enlightening to go back to our Janssen partnership as we have a phase 3, so we're obviously preparing for a potential BLA and potential commercialization, to actually look at potential commercial forecasts and go, wow, QC in the world can't supply this. So I would say right now, that is something that we really need to address.
Right. That makes sense. Okay. And then in the last few minutes, I also wanted to get your perspective on what you think people or investors might be missing regarding Meira's CMC capabilities.
Those poor investors out there at the moment, I think they're missing a lot about Meira in general. I do think that how close we are, the fact that we're in a pivotal, it's more than for AAV, it's more than we've supplied a clinical study. Because we're in a pivotal with our partners, I think that really shows that our entire manufacturing infrastructure is something that we're preparing for commercialization. And that is a pretty big task. And there aren't that many companies that are really in that later stage position. We're very lucky to have had the partnership with Janssen, the financial support, and the focus on this much later stage of manufacturing to get us there. I think that's missed. I think there's a lot missed in Meira, though, not just the CMC.
Absolutely. So, you talked about the late stage manufacturing, and we'll touch on this more in the upcoming panel regarding the evolving standards in gene therapy. But can you maybe give us a preview or a sense of the key lessons that you've learned regarding that manufacturing as you advanced your programs through the clinic?
Yeah, there's a lot said about the FDA's standards are higher than they are for other therapies. I'm not sure if that is true. I think that the FDA has an obligation to make sure that any product is safe and reliably produced. And that's quite hard to show in gene therapy and cell therapy today. So I don't know that the FDA's requirements have got stricter, but I do think that more of the industry is getting later stage. And so coming to a point where you really have to take those requirements for safety and consistency batch after batch very seriously. So I don't know that the FDA has changed so much, but more of us are getting to the point that we are at Meira. I mean, we've filed CMC amendments with 10 or 15 global regulatory agencies in the last, 6-9 months. And I don't think that we're receiving questions that are sort of onerously strange. I just think that as you get to those late stage development, you know, that's what you have to do to have a product that is given to people in the market. It just is.
Right. Mm-hmm. Great. So I think we're at the end of our time for this session. So I'd like to thank you, Zandy, again for your participation, the great discussion, and we'll chat again at 12 for the panel regarding the standards in gene therapy.
Thank you, Gula. Speak to you later.